is Abergas, and I am going to recite the genetic DNA translation, transcription, and translation. DNA replication has 4 steps. Step 1 is replication, Step 2 is binding, Step 3 is elongation, and 4 is termination. A replication fork is formed by the opening of origin of replication. A helicase separates the DNA strand. An RNA primer is synthesized by primase and elongated by DNA polymerase. Step 2. Once the DNA is separated, an RNA called primer binds the free end of the strand. Step 3. Elongation. During elongation, an enzyme called DNA polymerase adds DNA nucleotide to the free end of the newly synthesized polynucleotide strand. Step 4. Termination. Once both continuous and discontinuous strands are formed, an enzyme called exonuclease removes all RNA primers from the original strand. DNA replication enzymes. There are 6 enzymes in DNA replication. DNA helicase unwinds and separates double-stranded DNA as it moves along the DNA. DNA primase, a type of RNA polymerase that generates RNA primers. DNA polymerase synthesizes new DNA molecules by adding nucleotides to leading and lagging DNA strands. DNA gyrase unwinds and rewinds DNA strands to prevent the DNA from becoming tangled. Exonuclease are a group of enzymes that remove nucleotide bases from the end of a DNA chain. DNA ligase joins DNA fragments together by forming phosphodiester bonds between nucleotides. In DNA replication, there are 5 factors. Enzyme. An enzyme is required for the covalent joining of the nucleotide to the primer. Primer. The enzyme that synthesizes the DNA polymerase. Template. A nucleotide that is to be incorporated with the growing DNA chain is to be selected by base pairing with a template strand of DNA. Substrates are 4-deoxyribonucleotide triphosphates that are required for synthesis. DNA transcription has 3 steps, which is elongation, initiation, and termination. Step 1. Initiation. This is the beginning of the transcription. It occurs when an enzyme called RNA polymerase binds to a gene called the promoter. This sends a signal to the DNA to unwind so the enzymes can read the bases in one of the DNA strand. Step 2. Elongation is the addition of nucleotides to the mRNA strand. The RNA polymerase reads the unwound DNA strand and builds mRNA molecules using the complementary base pairs. Step 3. Termination. It is the end of transcription. It occurs when RNA polymerase crosses a stop sequence in the gene. The mRNA is complete and detaches itself from the DNA. DNA transcription enzymes. The main enzyme used in DNA transcription is RNA polymerase, which uses a single-stranded DNA template to synthesize a complementary strand of DNA. DNA transcription factors. The transcription factors involved are proteins that are involved from transcribing DNA into RNA. Just like DNA transcription, DNA translation also has 3 steps. Step 1. Initiation. Recognition of starting codons and binding of ribosomal mtRNA at the P site. Step 2. Elongation, forming of peptide bond and growing of polypeptide chain. Step 3. Termination. Stop codon enter the A site, resulting in the termination of the process. Pepsidyl transferase is the main enzyme used in translation. It is found in the ribosomes with an enzymatic activity that catalyzes the formation of covalent peptide bonds between the adjacent amino acids.